Are we live? Okay. Good morning and welcome again to the third day of the World Congress for Brain Mapping, our 18th annual. Today we are delighted to have Professor Andre Machado from Cleveland Clinic to give a lecture. And Professor Machado has recently joined our board, and without further delay, I would like to welcome Professor Machado.
Thank you. Can you guys hear me okay? It's a pleasure to be here today. I want to thank you so much for this very kind invitation. I wish I could be there in person, but I couldn't make it work. So thank you for including me by video connection. What I want to talk about today is a 10-year project that we are developing here at Cleveland Clinic to advance the use of deep brain stimulation for post-stroke rehabilitation, to promote the rehabilitation of patients that have been affected by a severe stroke with significant hemiparesis. I have adapted this presentation to meet the objectives of the Brain Mapping and Therapeutic Society. And I'm going to try to make a link as we go through this of how brain mapping clearly can link and lead to a novel therapy. The disclosure slides here to begin. We have funding from 3 NIH sources, in particular the Brain Initiative, that have supported this work. There is a conflict of interest with Inspire, which is a spin-off of Cleveland Clinic, and I will talk about off-label use of a medical device. I don't believe I need to persuade this audience that stroke is common and devastating, leading cause of long-term physical disability in the United States, in the industrialized world, and even in the developing world. Stroke is gaining a much greater level of importance in terms of public health as lifestyles change and as the developing world begins to get more and more developed. Disease also begins to look a little bit more like disease in the developed world. The idea of utilizing neuromodulation to help people recover from stroke is not new. And there are many types of neuromodulation technology that have been and are currently being evaluated for that goal. Going from less invasive to more invasive, left to right, we have transcranial magnetic and transcranial direct current stimulation, a very robust and rich literature that has taught us a lot about the physiology of the normal motor system and the physiology of the motor system during recovery. Cortical stimulation applied directly to the dura or to the surface of the cortex itself, subdural, is another form and has been also investigated. We are working with deep brain stimulation, placing an electrode in the depth of the brain with the objective of directly modulating a given network. And I will tell you what our hypothesis is. We have proposed some 15 years ago that modulation of this pathway that you see here, the dentatothalamocortical pathway, is a viable target to promote rehabilitation after stroke. What is it that we are trying to achieve? We're trying to utilize a node, a compact point, in a vast network to modulate a large region of cortex that ranges from prefrontal cortex all the way to parietal areas, meaning the cortex that is preserved around a typical middle cerebral artery stroke. And by modulating this network, we plan to reverse the deleterious effects of cross-cerebellar diaschesis and facilitate motor recovery by means of stimulating an excitatory glutamatergic network that has really two neurons between the place where we are placing our electrode, that's the dentate nucleus output, all the way to the cortical areas, dentate, thalamus, and cortex. We began our investigation in brain mapping. We began our investigation in a rodent model of normal physiology, followed by a rodent model of stroke, to evaluate what would be the frequency of stimulation that would promote the desired effects. For those of you that work in deep brain stimulation for Parkinson's disease, essential tremor, those are the standard indications. We typically use high frequency stimulation, defined as more than 100 pulses per second, more than 100 hertz. We didn't know if high frequency stimulation would be the way to go here. And we did a dose titration study, and we learned that for this pathway, the best and most prolonged excitation that we could achieve was at 30 hertz, either continuously or in a bursting fashion, and I'll show that to you in a moment. Then we applied that knowledge to a preclinical model of stroke, of cortical ischemia, and then we evaluated the effects of long-term 30 hertz stimulation in post-ischemia animals. And we evaluated that using an assay called the pasta matrix assay, which is shown on the left bottom corner of the screen. You can see one of our rats reaching for pasta. It has a certain tensile resistance. And the more they can break, the greater their recovery. That's the scale we use. And here is the result of one of our preclinical experiments showing the difference between stimulation plus physical rehabilitation versus physical rehabilitation without stimulation, red versus blue. And over the span of 5 weeks, you can see how the groups separate from each other. In a group that actually started with the same level of impairment, same level of severity, because they were severity matched. And we have been able to replicate this behavioral improvement, this improvement in motor function in the rodent a few times in our lab. It has now been independently replicated at the university, at Stanford University, with optogenetic stimulation, and has been replicated by an Asian group as well with electrical stimulation. We think that this is a viable and reproducible phenomenon. What about the mechanisms? What happens to the brain when these animals are recovering their motor function? What's underlying this recovery? Well, that's where brain mapping comes in. We performed extensive mapping of the perilegional cerebral cortex with intracortical microstimulation placed at 1500 microns of depth. That's the layer 5 of the cerebral cortex of the rat. And you can see on the far left of the screen the grid, the template that we used, and descended a microelectrode to map out the brain in each of these spots. And what you see on the second to the left, the naive animal, that is the normal motor representation of the rodent brain, where the yellow shows the forepaw, the hand, if you will. And you see that it is not a simple motor representation. This is the typical finding of a primary and a secondary motor representation of the rat. It has been thoroughly published in the literature. Then one to the right, you see sham, meaning the animal suffered a stroke, but we didn't treat with electrical stimulation. And you can see that the representation of the paw is gone, meaning we successfully caused a stroke in the animal, and there was a hemiparesis to follow. Then you see two types of stimulation, burst and regular, and you see a resurgence of the representation of the paw. By no means a normal representation, yet a new representation that is correlated with the improvements that the animals experience. What about markers of long-term potentiation? This is a busy slide. I'm going to direct you to the column, the left side column that says perilesional. And you see on the far left where we obtain the tissue from. We obtain the tissue from the perilesional area, and then contralateral and the visual cortex, those are controls. But if we look at the perilesional cortex and we see the expression of synaptophysin, that is the bottom left of the screen. I'm going to try to draw around that to show you the highlight of it. And you see the blue mark that I made here, showing that synaptophysin, when stimulated, that the expression of synaptophysin went back to above naive levels. Synaptophysin is a protein that occurs only in the synaptic vesicles, in the presynaptic vesicles. So this means that there is a significantly greater expression of the proteins that are making the synaptic vesicles with stimulation, which suggests synaptogenesis. Now, of course, we don't know if synaptogenesis is being attempted or if it's being achieved. And to address that, we utilized a different type of assay, which is going to be in the next slide. And that is the utilization of three-dimensional electron microscopy, shown here, of three-dimensional electron microscopy, shown here on the left side. I'm going to try to highlight this here with a cursor. What you see here in panel F is the post-processing coloring. The coloring is not part of the picture. We did that only for explanation purposes. And you see in yellow the presynaptic, you see in yellow the postsynaptic component. In blue, you see the presynaptic component, and you can even see the little dots representing the presynaptic vesicle. That is three-dimensional electron microscopy. And the black line is the postsynaptic density, which is what we really use to indicate whether the synapse is fully formed or not. So with this technology, we were able to then count the number of fully formed synapses. And what you see is that comparing stimulated animals versus non-stimulated animals, that there is a doubling of the number of synapses in the perilesional cortex, going parallel with the recovery experienced by the animals. So if we were to summarize this finding so far, what we have is an emerging therapy that has shown to be effective to produce motor rehabilitation that is associated with improvements in expression of markers of long-term potentiation and synaptogenesis, and is also associated with reorganization of the perilesional cortex. Well, why am I showing you this picture again then? Because I want to go to human translation in a moment, and I would like you to remember this picture here that shows the on-off, on-off, on-off demonstration that stimulation also produces increments in perilesional excitability. And here we have transitioned from the small brain of the rodent to a macro electrode, a DBS electrode placed in the dentate nucleus of the first humans. This is a first-in-man study. And what you see here is an experiment conducted by one of my colleagues, Dr. Ila Plau. She would be a great member of this society. Maybe she's already a member of this society. And she's conducting pretty much the same experiment that we conducted in rodents to evaluate perilesional excitability now in humans. Of course, we're not placing a microelectrode in the layer 5 of the motor cortex to measure excitability in humans. We're using transcranial magnetic stimulation to measure excitability. So the patient has an electrode placed in the dentate nucleus of the cerebellum and is undergoing TMS. The TMS is measuring the excitability, and the DBS is modulating the excitability. And what you see here is that when we compare the effects of active DBS versus sham, meaning the patient thinks it's on, but it's not, and here's the clicking of the TMS, here's the scissors programming the DBS, right? The patient is blinded. We see that there is a reduction in the threshold necessary to produce a motor evoked potential, and there is an increment in the magnitude of the motor evoked potential associated with a TMS pulse that we do not see when we are evaluating the excitability with sham deep brain stimulation. So essentially what this slide here is showing is that we were able to reproduce in humans the changes in excitability that we had seen in the rodent model. DBS in humans modulates perilesional excitability. What about closed loop? Can we record from the dentate nucleus and then use that to one day potentially do something that will be more automated? That's possible. So let's go back. We were transitioning from rodents to humans. Let's go back from humans to rodents to do a little brain mapping. We placed a grid of 5 microelectrodes into the equivalent of the dentate nucleus of the rat. Then we placed this rat in front of a task that video records the animal as it's reaching for a sugar pellet, as we're feeding the animal. And when the animal reaches, it breaks a beam, and that allows us to time sync the movement of the animal with the activity with the brain recordings. And we evaluated the videos to see if the reach to get to the sugar pellet was successful, that's a hit. Or if the animal missed, if the animal accidentally dropped the pellet, or if it had to use more than one strategy to grab the sugar reward. And here is the local field potential findings from this array of microelectrodes. I am going to try to get back to my annotate mode here because I want to show you the dotted line. That is the moment when the beam breaks. That's the 0 moment. That's the beam break. And you see the power spectra of the findings. What do you see here? Have you noted that there is substantial activity in the beta band before the animal actually, before the animal hits the beam, before it breaks the beam? Why is that? It's because the cerebellum gets a copy of the motor plan from the cortex, and it knows that the motor plan is bad before movement even begins. That's when the cerebellum is trying to make compensatory measures to change the motor execution. It's even before the execution of the movement begins, telling you how important the cerebellothalamocortical pathway is for motor planning and motor correction. And we think we'll be able to explore this finding later to be able to find motor plans that will be poorly designed in patients recovering from stroke and stimulate in a way to make them more effective. I want you to remember this slide because we're coming back to this slide again in a moment when we go back to humans. Which we're doing now again. So now we are looking at a task in which one of our patients that has the DBS electrode already fully implanted, the generator is inside the chest, typical DBS setup. And what you see in this video here on the right side of the screen is what the patient sees. The patient is asked to use a dynamometer, a squeeze tool, to follow the ball. When the patient sees squeeze, they're supposed to squeeze and release on cue, or squeeze and hold, and then release based on this visual feedback. You see here the electrode tip co-registered to the dentate nucleus. On the left side of the screen, you see it relative to the skull. And now we're going to observe what happens when the patient is performing this task by either the passive recordings of the DBS lead. This is a local field potential from the DBS lead itself, or when we turn on the DBS, and we're going to look at the effects in the cortex. So what you see here are the 8 channels of the DBS lead. It has 8 electrodes, from 1 to 2 on the bottom right corner to 7 and 8 on the upper left corner. Can you see that the orange line, which represents the recordings done when the patient was performing the activity with the affected hand, you see that there is a significant gain in local field potentials on the electrodes 6 and 8 and 7 and 8. This is an indication of the level of activity in those electrodes, which is an indication that these electrodes are positioned in a spot in the dentate nucleus that is relevant to motor execution and may be viable for programming. If we now look at the desynchronization that happens at the cortical level when we compare stimulation off versus stimulation on, we see the results of our programming of this device. On the upper line, the top line, you see DBS off and on, and there is not much a difference at all. DBS in those settings that we selected did not produce a significant change. If you go to the bottom 2 panels, when you compare off and on, now you see a very significant change in the power spectra, particularly in the lower frequencies, around 10 and 15 hertz. And this change in the event-related response at the level of the cortex was associated with motor gains, meaning when we programmed DBS chronically to the second setting, not to the first setting, we saw robust improvements in motor function. In the interest of time, I'm going to go to the clinical trial, and I'm going to show you the summary of our results. So when we implanted and programmed and chronically stimulated patients who were at least 1 year post-stroke, between 1 and 3 years post-stroke, here is what we found. We found that for all patients, there was at least some improvement in motor function. The definition of response here, of a meaningful improvement in motor function, is at least 5 points. And you see on the y-axis the scale. So for patients that have no motor function left at all after stroke, there is improvement, and 2 patients out of 5 actually responded, got to that threshold of at least 5 points. But when you look at patients that had at least a minimum amount of residual function at the time of enrollment, then we see that there is a robust and consistent achievement of this minimum amount of recovery, in some cases more than doubling the recovery that is considered to be minimal. What does it mean to the patient? I'll give you a couple of quotes here. I wasn't able to pick up a pen, but now I can write. She couldn't write cursive, but she can use uppercase. She can actually work a pen. I wasn't able to close my pants and zip up my pants. Now I am. My left side used to be just a dead appendage of my body, and now, even though it's still not normal, I can use that left side to work the tools that I need to work as a car mechanic. It's a supporting hand that went from being an appendage to being a useful limb. In terms of complications, this was very well tolerated. There were no major perioperative complications. We had nausea in the first 3 patients, which were able to resolve in the last 9 patients. And I'm going to close with this video here from our first patient. Her name is Judy. We're allowed to use her name. And what she's doing here is that she's pitching the first ball of the Cleveland Indians baseball team. She's opening the game. And what matters here is not that she can grab the ball. What matters is that she can open her hand and release it on time, which was completely impossible for her before this treatment began. In the end, as a translational group, all this brain mapping that we have done really is summarized by the recovery of these patients. This is what we care about. And we think that we are on a path that is at least encouraging, and we would like to pursue this into a phase 2 clinical study in the future. So for a summary, the effects seem to translate well from rodent to human. The surgery is well tolerated. Patients with at least minimal motor preservation at the time of enrollment do quite well in the study, and programming is guided by electrophysiological biomarking. These are my colleagues that I want to thank, in particular Ken Baker, who is my partner in this study. Thank you very much.